Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about another test that name is deoxyribonuclease test. For that test we mostly used a media which we called it deoxyribonuclease agar media, which can contain DNA molecules. Okay. Now let's talk this test with their introduction. As all of you better know that DNA is a large polymer and the cell cannot take it. So for that purpose the cell have to degrade it into nucleotides, because all of you better know that DNA are made up of nucleotide. So once the cell degrade it or break down it into nucleotides, so then the cell will easily take it. So for that purpose there are some bacteria that can secrete exoenzyme which we called it DNase. By using this enzyme, what actually happens, the bacteria will degrade the DNA into nucleotides, and after that the bacteria take up these nucleotides by a process which we call it cell membrane transport process. So through this way the protein or the nucleotides will be easily uptake by the cell. Now the cell use these types of the nucleotides to make their own nucleic acid, and they can use these steps of the nucleotide as a source of nitrogen, phosphate and carbon. So this is a little bit about DNase test agar media. Now let's talk about their uses, that how we can use this media. So this media is specially used to identify the bacteria that can produce exoenzymes, or we can say DNase enzyme. Okay. Moreover, this media is used to determine the ability of an organism that can hydrolyze the deoxyribonuclease by exoenzyme. Then we have some strain like Staphylococcus aureus and Staph epidermis. So using these types of media we can easily identify the Staph aureus and Staph epidermis, because we better know that the Staph aureus is actually producing the DNase enzyme, while the Staph epidermis do not use the DNase enzyme. Do not produce the DNase enzyme. If we do not have the plasma, so for that purpose we mostly perform the DNase test media. Moreover, this coagulase test can also give false type of the result. That's why using this test it can give accurate result. Now next point says that we can also use this type of the test for the for the identification of Escherichia, that is positive DNase, and Enterobacter, that is negative DNase. Moreover, we can also use this type of media, this type of the test for the Moraxella catarrhalis, that is positive, and Neisseria, that is negative. We can remember guys, in this case although the MRSA strain belong to the Staphylococcus aureus, but remember guys, the MRSA strain do not produce we can say that DNase enzyme. So for that purpose it will give negative result. Okay, so for this purpose how we can identify the MRSA? So we can identify it by using coagulase test. Okay, and there are some other strain like Staphylococcus capitis is also using it can also give a weak positive result. Now let's talk about the composition of this media which are used for the DNase test. It composed of pancreatic digest of casein, pipic digest of soybean meal, sodium chloride, distal water, deoxyribonucleotide or deoxyribonucleic acid, and then we have agar. So these are the amount of this ingredient, as you can see. Now let's talk the principle of this ingredient which we talked. So as we better know that DNase are what? These are the extracellular endonuclease that can cleave the DNA, and once the DNA are cleaved, so then we get nucleotide as well as phosphate. So now the DNase agar media which we are used for the DNase test, it contain methyl green, which is acting as an indicator, green indicator. Okay, so remember guys, when methyl green is a cation and when it binds, so then it is negatively charged DNA. So methyl green is a cation which binds to negatively charged DNA. How we can identify the bacteria which can be creating DNase enzyme? So remember guys, in this case when the DNA are broken down, so what happen, it will binds to this methyl green. After that what we get a clear hollow zone where we streak our bacteria around that. Okay, that can showing that the production or the release of DNase enzyme. If the DNase agar do not contain methyl green, so then we use HCl. Okay, so after streaking in inoculation after 24-hour incubation, so then we use HCl. So by using HCl we can also identify the positive and negative reaction as well. One thing more, the presence of the deoxyribonucleotide or deoxyribonucleic acid outside the membrane, so it will specially used to detect the enzyme. So once the enzyme is released, so then it can easily be identified on the media. Okay, so that's a little bit about the principle. Now how we can prepare this media, let's talk about that. First of all, suspend 42 gram of the media into 1 liter distilled water. Heat, mix it and dissolve. After that, do not autoclave it, because once you autoclave, so all the ingredient and nucleotide will be break down. Then cool down this media and pour it into petri plate, sterilized petri plate. After that, take up the bacterial colony using a sterilized wire loop and streak it into DNase agar medium. Then incubate it at 34 to 37 degrees Celsius for 24 hours. After incubation, check the color changes in the methyl green area, which can show the DNase enzyme. Then if the methyl green are not used in this media, so then for flood this agar medium with 1% HCl solution. After that, observe the absorption into the plate. That can show the positive result. So this is all about the preparation and inoculation of this test. Now what type of result we get on this media? So remember guys, if we want to see the positive result, so mostly the medium is colorless around the test organism, and the positive bacteria are Escherichia marcegine, Staphylococcus aureus, Campylobacter jejunum. The negative result will be that DNAs are not degraded, and due to this way the medium remain green, and the organism of the negative are Staphylococcus epidermis and Neisseria gonorrhea. Then these are the picture. As you can see, this A1 showing the positive result, B is also showing the positive result, while the C is showing the negative result, and this green can showing the methyl green indicators. Now this is the media without methyl green indicator. So for that purpose, once the inoculation has done, after 24 hours we add what? HCl, and then we wait for 5 to 6 minutes. Once the HCl are absorbed, as we can see, so then it means that DNase enzyme are secreted and it will degrade the deoxyribonucleic acid molecules that is present in the media. So that's all about the DNase test agar. Thank you so much for watching.